Hello, listeners. Welcome to this episode of Cell and Gene, the podcast. My guest for this episode needs no introduction, but I will give you just a glimpse of his robust background. Dr. Bruce Levine is the Barbara and Edward Netter Professor in Cancer Gene Therapy at the Perelman School of Medicine at the University of Pennsylvania, and he is also ISCT's immediate past president. He is the founding director, Clinical Cell and Vaccine Production Facility, or CVPF. He is a leading innovator in the cell and gene therapy space, as he is known for his groundbreaking work in the development and use of synthetic immune cells, or CAR T cells, to attack advanced cancer cells. And if you look up his bio on the Perelman School of Medicine, you will see the impressive and extensive list of awards and honors he's received, as well as just some of his publications. Bruce was my guest on episode 3 of Cell and Gene, the podcast, so please do go back and give that one a listen. It's chock full of really great information. And so I'm thankful to have him back to talk to us this time about his time as president at ISCT, as well as progress in CAR T and a few other things. So Bruce, thanks for being here on Cell and Gene, the podcast. Welcome back.
Great to be with you, Erin, and congratulations on the 70 intervening episodes. I know you've had some great people on, and as evidence of the importance of what you're doing, to be able to continue it.
I really appreciate that. That means a lot coming from you, so thank you for that. Well, let's jump right into it. So you are ISCT's immediate past president. Now, I'd love to hear in your own words, during your time as president of ISCT, what were some of the things that you and your team at ISCT, and including its impressive membership, right, that you were able to bring back to Penn and employ in your role there?
Yeah, well, going back to 2018, when I became president-elect for 2 years, so we have a two-year system at ISCT. Our membership in 2018 was 1,450 and going up, but it had fluctuated a little bit in the past years. This year, we will be in the upper 3,000s in terms of membership, so more than doubling. So tremendous growth, and the new members see the value of ISCT. Now, I had a vision at that time of investing in our mentoring program and in our workforce development and education programs, and that's now become the ISCT Institute of Training and Development that's led by Raymond Lamb. That institute offers more than a dozen courses and programs ranging from lab bench to regulatory. So it is global as well, as is ISCT. My vision when I came in also was communicating the translational space that ISCT was in, but also our 3 domains, and thinking of it like the Rosetta Stone, right? So you had 3 languages, and you had to be proficient. You have to be proficient in 3 languages to move cell and gene therapies forward. So what are those? Well, we have a pillar based in academia that's science-based. We have a pillar that's based in industry that is commercialization-based. We have a pillar in regulatory and quality operations. So you need to understand all of those to bring cell and gene therapies from bench to the bedside to the patient. I also want to acknowledge what has come with our profile and our tremendous growth is that we provide advice. We have KOLs that provide advice and expertise to not only to our members, but to government agencies, to NGOs on cell and gene therapies. And so we're seen as that trusted voice. And finally, I'd like to acknowledge in ISCT the leadership, my predecessor, John Rasco, Jacques Galipeau, who's current president and will take my place as immediate past president, and especially Queenie Zhang, who's led ISCT as CEO. It's been an honor and a pleasure to work with her, and I've learned so much from her on on strategic thinking and the ins and outs of the society and how we position ourselves for growth as the field has grown tremendously over the past few years.
Absolutely. And speaking of that tremendous growth in the field, it's almost, if from my point of view, what we do at Cell and Gene, uh, it's in a good way hard to keep up with the growth that's happening. So I suspect that more of that will be covered in the upcoming ISCT, which is this in Vancouver. And I wanted to get your take on that. So I know you're a speaker at this year's event, and I certainly don't want you to, give away the store in terms of what you'll be covering. We're recording this in advance of the ISCT event, but was hoping you could share maybe just a sentence or two about what you're covering, the format, and what are you, as the immediate past president, what are you most excited about for this year's meeting?
Well, this will actually be our 30th annual meeting. So we have a celebration for that. And we expect it to be our biggest meeting yet. Now, we made some revisions to the way the meeting was organized a few years ago, and I think it's worked out really well. So we have a translational pathway program that includes all the plenaries from the presidential, but then the linear progress of cell and gene therapy. So there's one on science and innovation, then translation to the clinic, then from first in human to clinical acceleration, and finally launch and patient access. And so that flows along as the days of the meeting flow along. A couple of years ago, we added a new format called a roundtable, and that was designed to be immersive, highly interactive, maybe some debating and focus group discussions to drive and impact advancements in the field. So those are not didactic, fully baked presentations, but it's taking a topic and saying, well, here's my experience and what would you do? Well, what would you do? What does this mean? I also want to, I guess, highlight some sessions that, for me, either I'm chairing or speaking or of interest to me. In the translational pathway, we have a session, the cutting edge of stem cell innovation. We have another on enabling gene editing platforms for novel in vivo and ex vivo therapeutics, and a third that I'm interested in are in vivo cell therapies, the future. And then as far as roundtables, there's one on autoimmune diseases. That's a very hot topic. It seems every CAR T company or other companies are going into autoimmune diseases these days, addressing the FDA announcement in November of their investigation on secondary malignancies. Are CAR T cell safe? Then investigating the risk in the context, and then in terms of ethics, we know that there are direct consumer marketing of unregulated cell and gene therapies. There's a roundtable on emerging shadow markets and cell and gene therapies. Can you protect your patients? And these are not just stem cell, quote unquote, stem cell therapies. They're now exosome therapies. They're now oligonucleotide derived therapies or so-called therapies being marketed directly to patients. So that is unregulated, unethical, unfair, and danger to patients, not just for their health, but to their wallet. And we'll have a discussion on that. So very much looking forward to that and all the content we'll have throughout the week at the meeting.
Yeah, it's not, no surprise here that it sounds like it's going to be a really jam-packed, robust schedule with all sorts of formats for the audience members who want to be there. I would love to, on the back end of this, come back and talk about what was covered and why during the session on the unethical approaches to the gene and cell therapies that you're referring to. Um, I just think there's so much to be learned, and our audience especially would love to know, uh, you know, some of the hopefully potential solutions that come out of that discussion to kind of mitigate that problem. I would love to talk to you on the back end of that, as well as certainly, like you said, the autoimmune portion of this. Even later this year, we're doing a live event, and that actually is the topic. So to your point, it's we weren't talking about this 5 years ago, and then here we are. It's a major topic that we're covering. So I think the ISCT event in Vancouver this year is going to be you're going to come out a lot smarter than you would when you went in, for sure.
Yeah, yeah.
Okay, I want to talk a little bit about moving on to CAR T therapy. So I don't think I need to tell my listeners this, but just to bring them up to speed, you pioneered the first FDA CAR T immunotherapy treatment for leukemia and lymphoma. So being the co-inventor of Kymriah, so which was then licensed to Novartis. That was years ago. Here we are today. Talk to us about the current landscape for CAR T therapy. What has progressed in that time since the initial approval of Kymriah? And, what does the near-term future look like for CAR T therapy?
Yeah, so Kymriah was approved in 2017. About 6 weeks later, Yescarta was approved, and we have 4 more CAR T products approved now in lymphoma, leukemia, acute lymphoid leukemia, and myeloma. We also have 4 approved products in China in leukemia, lymphoma, and myeloma. Altogether, I estimate about 40,000 patients, probably more, have been treated with CAR T cell therapies. We know that from media interviews following the announcement of the FDA investigation in November, December, where the companies, if you total them up, it's 34,400. That doesn't include China, doesn't include clinical trials, and doesn't include the intervening 3 months. And speaking of by 3 months and quarter, if you look at the sales of those therapies, it adds up to $1 billion per quarter.
Wow.
So $4 billion a year. That doesn't include China. And yet, only 20% of patients, as estimated by Christy Shaw, only about 20% of patients who could possibly receive CAR T, where they're eligible, will receive it. So that means that we have inequities. We have inequities in education of patients, of oncologists, access in rural areas of jurisdictions where we have approvals like the US. And then we have many countries around the world that don't have access to CAR T. As far as I can tell, in Africa, there may be 1 or 2 trials or maybe 0 clinical trials, and certainly no commercial CAR T available. So we need to work on that, providing access for the therapies that target CD19 and BCMA where we have approvals. We also, I've said, maybe I said in your earlier podcast, that we need to work on solid tumors. Well, we have been in the intervening time, and we're having some successes. So we have indications of clinical activity in glioblastoma or brain cancer. Just a few weeks ago, within 7 days, there were 3 significant reports from 3 separate institutions, Penn, City of Hope, and Mass General, showing activity with direct administration to the brain of CAR T cells. We're also seeing success in some improvements we've made in potency going back to blood cancers, amending the manufacturing, shortening it to 3 days, and implementing in the same construct an anti-tumor cytokine IL-18. And so what we've seen with the eligibility in that trial being patients, if they had been eligible to receive a commercially approved CAR T, had to have received that and failed. So we're taking those patients, enrolling them, treating them, and putting the majority of them into a remission. So think about that. They failed a CAR T. Now they're getting this next generation, shortened manufacturing, integrating IL-18, and we're putting them into remission. So we're very excited about that and the other progress that's being made. And then thinking forward and getting back to part of your question is, what's next and how can we address the fact that only 20% of patients receive CAR T? We have very exciting data, clinical trial data in autoimmune disease. What happens when we have approvals in autoimmune disease and solid cancers and the blood cancers? There aren't enough humans on Earth to manufacture those ex vivo therapies. So I'm co-founder of a spin-out developing in vivo CAR T. There are about a dozen other companies in the US and around the world looking at in vivo CAR T using either a lentiviral vector or a virus-like particle or a lipid nanoparticle encapsulating messenger RNA. So think of COVID vaccines marrying CAR T cells, and that's how you would describe that approach to your friends and family. So as you said up top, it's hard to keep up with everything that's happening. I devote a significant amount of time to going through the announcements, reading papers, and trying to keep up in the field.
Absolutely. And, when you look back when you started years ago, you partnered with Carl June, and even before that, did you ever think, cut to 2024, some portion of years down the road, in your exceptionally robust career, did you ever really think we would see this level of success? And I know there's we have room to grow, but just looking back, how remarkable it must be to see what has been able to come to fruition today, and what's now possible, what probably one day people didn't think was possible.
Yeah, I think, well, first, I need to acknowledge and emphasize that I'm part of a very large team here at the Center for Cellular Immunotherapies, led by Carl June. We have about 230, 240 people here in the center. Our manufacturing facility, we have about 50 people there. And there are other groups working on CAR T. And yeah, the profile, just this past weekend, Carl June and Michelle Sutherland received the Breakthrough Prize in California. That's called the Oscars of Science for all the celebrities that attend. So there's recognition all over the world in different domains, Hollywood, wherever, of cell and gene therapy. So it is remarkable, but I keep coming back to the patients and knowing that we've had an impact there and being connected to the patients. I correspond with our earliest patients, the adults and the kids. And Emily Whitehead was the first child in the world treated with CAR T. And just over there, a few hundred yards, she's a freshman at the University of Pennsylvania. So that's crazy to think as well.
It's wonderful. All of it is just it's so promising, and but to your point that the patients are the ones who get the real benefits. Emily included, and everyone else was a recipient of CAR T therapy. Well, speaking of Promo Center, University of Pennsylvania, right out your window there. And I'm just down the road a little bit. Last time we talked, of course, we discussed Cellicon Valley, the term given to Philadelphia due to its hub status in cell and gene therapy. Again, talk to us in your own words about Cellicon Valley and how it's progressed in the last few years, and what's in store for Philadelphia in terms of academia and industry and the partnership, and you know, why it is Cellicon Valley, and how, what we're looking at maybe sample years.
Well, somewhere around 2012, I guess it was, I had Emily's parents, Tom and Carrie, in my office, and I was trying to describe to them what we were building at Penn and in Philadelphia, writ large. And I said, well, it's kind of like Silicon Valley. And it just kind of popped into my head, and it made sense because it's new technology and now other people are using it, which is great. And there is a big emphasis on cell and gene therapy in this region. If you count cell, gene, and nucleotide-based therapeutic companies, there are more than 55 of those in this region, according to the CEO Council for Growth. If you look at new jobs that have been created, they've doubled since 2019 to more than 10,500 jobs in cell and gene therapy and connected health. And there's building of labs, startup space, GMP space. We have plans at the Pennovation Center over on Grace Ferry Avenue to build several 100,000 square feet of cell and gene therapy and related space that will include process development, manufacturing, analytics, and all kinds of related activities. We have meetings that are attended by folks from around the world. Every 2 years, we have a Silicon Valley meeting. This year, we called it the Future of Cell and Gene Therapies. We'll do that again in 2025. We've been selected by the ISTCT and ISCT 2 cycles in a row, and I hope a third, for the cell therapy training course. That's a week-long course for selected scholars from around the world. And I mentioned our Center for Cellular Immunotherapies. We have a new RNA Institute led by Drew Weissman, Nobel Prize winner. We have activities in cell and gene at the engineering school and at the vet school, where we run CAR T-cell clinical trials for dogs. And we have spin-outs from Penn, from our center. I mentioned the overall that we have in the region. So I think it's an exciting place to be, maybe the place to be if you're in cell and gene. And I can tell you that the caliber of students, fellows, med students, grad students at all levels applying to Penn, seeking to come to Penn, has increased, so that I think we have the cream of the crop here now. So wonderfully talented early stage professionals here.
Oh, I couldn't agree more. And the best part is they can stay. They don't, everything they need to continue on with their research and their profession in whatever capacity they choose is right in their backyard. They can simply stay in the area and continue on to do really good things for the patient. So I couldn't agree more. I'm so happy that it continues to progress and improve as the years go on, and that Cellicon Valley has stuck. I know it was a term you kind of coined in that meeting with Emily's parents, but imagine that it turned into its own meeting, and because Philadelphia is, like you said, the hub for cell and gene therapy. So it's great. Um, I earlier on you mentioned the startup that you co-founded. I just wanted to make sure our listeners know what that is.
Yeah, so there's actually two startups that I'm a co-founder of. One is Timidity, that was recently acquired by Kite Pharma,
Mhm.
and they're developing clinical trial technology and CAR T cells that we have here at Penn. The other is Capstan Therapeutics. So co-founders of that include Drew Weissman, Carl June, John Epstein, Ellen Pure, Stephen Albelda, and Hamida Porteous, and Egad Kajanian, who's now at the company. So they're based at San Diego, but have very close ties to Penn, and we're developing uh, mRNA encapsulated by lipid nanoparticles. And the key here is that they are targeted to T cells by an antibody or ligand that is attached to the outside of the lipid nanoparticle. Because if you don't have that, lipid nanoparticles will naturally traffic to the liver. Well, we don't want them, in our case, to go to the liver. We want them to go to T cells. So we have a ligand that does that. And we're seeing extraordinary demonstration of activity in preclinical models, not only in mice, but in non-human primates. And I think the company will be showing that data later this year at a forum to be determined.
Okay, well, at Cellicon, we'll certainly keep our eyes on that. We've absolutely covered both Capstan and Community in various ways. So on Cellicon for our readers, and we'll continue to do so. So looking forward to see what comes from both companies. Okay, well, that sort of brings us to the end of our formal discussion here. At the end of the episode, I like to talk to my guests about who they are when they're not developing or manufacturing life-saving therapies. And so when you were on last time, you talked a lot about how you were very involved in and had a passion for gravel biking. And I was wondering, cut to, probably 2 years later, are you still gravel biking? And if so, how's it going? If not, what have you moved on to?
Well, maybe I should explain for those who aren't familiar what gravel biking is. So you take a regular bike, a road bike or a hybrid bike, and you put some tires with a little bit of grip on them, not quite a mountain bike, a little bit thicker frame than a road bike, not as thick as a mountain bike, but that allows you to go off-road, like rails to trails, and stay away from the cars and see some scenery and lakes and trees and things that you ordinarily wouldn't be able to do on a road bike because it wouldn't be as comfortable. And so I was doing that and traveling a lot. And then in October, after I got back from a week in Dubai in India, attending meetings and speaking, I was scheduled to go with buddies on a ride. And while going down the stairs at home, I found myself on the floor and my kneecap hit the ceramic floor. And when that happens, it sounds like hammer on stone. So turns out I had a fractured patella, a ruptured quad tendon, and a partially torn muscle. So I had surgery for that. I was in a mobilizing brace for 2 months and have been doing physical therapy since then. I'm doing well, and I've been on my bike since March 1st, just very gingerly and low mileage at first, but it's actually good therapy. So I've gotten back out there, not fully recovered yet, but getting there. So my advice to your listeners is if you think you're feeling a twinge in your knee, well, it could be something happening. I think I had a partially ruptured tendon that just caused my knee to give way. So also hold on to those railings.
Oh, well, one, I'm so glad you're doing well on the other side of surgery and back up on the bike, uh, so to speak. But boy, I'll tell you what, I know what you mean. one day you're completely fine. You think no matter what, you're invincible, and then one wrong move and you're looking at surgery. So I'm glad to hear you're okay and that you're
Yeah.
talking about the
So
life.
I have a stainless steel souvenir in my niche that reminds me of that.
Oh, did you have your surgery at Penn?
Yes, yeah, at Penn Presbyterian by a retired U.S. Army trauma surgeon. So I'm sure he's seen worse than me. So I have a clamp holding the two pieces together, a hook on the top, and it's screwed in at the bottom. So I say I'm more screwed up than I used to be.
You have to have a sense of humor about it, or else, you know what I mean. We're all in, it's not good otherwise. But it sounds like you were in the best of hands with your surgery, and again, I'm just so glad you're on the other side of it. Um, all right, listeners. Well, that wraps up this episode of Cell and Gene, the podcast featuring Dr. Bruce Levine. Bruce, thank you so much yet again for your time and for the exceptional amount of insight you shared once again with our listeners. We're so happy to have you here. Do look forward to having you back again sometime soon.
Thank you, Aaron. Great to speak with you.
All right, listeners, thanks for tuning in, and we'll talk to you soon.